Australian National University's Research School of Chemistry. Dr. Huber received his diploma of chemistry from the Technical University of Munich and completed his PhD at ETH Zurich. He has since held positions in physical chemistry at ETH Zurich in 97, in the supercomputing facility at the Australian National University from 98 to 2000, in the Department of Mathematics at University of Queensland from 2001 to 2005, and the School of Molecular Biosciences at University of Queensland in 2006 to 2010. He moved to the Australian National University as an ARC Feature Fellow from 2010 to 2014, and before being appointed professor at the Research School of Chemistry. Dr. Huber specializes in computational chemistry, bioinformatics, and protein structure. He's particularly well known for developing methods to extract three-dimensional structural information from minimal or sparse experimental data, most notably using bioinformatics to infer structural information. Recent works have been particularly fruitful in encoding an application of fluorinated amino acids for F19 NMR work to resolve protein structure and conformational dynamics. His career is highlighted by an extraordinary publication record of at least 150 publications, according to his Google Scholar page. But the most impressive statistic I could find wasn't the number of publications, but the number of citations his work has garnered. John, can you guess how many citations Dr. Huber's most cited paper has?
Oh, this is a trap. I'll go 3,000.
12,588.
It's an outlier.
That's an outlier? Oh, I was hoping that was going to be. Oh man, okay, all right. So anyway,
that's impressive nonetheless.
Nevertheless, extremely impressive a career of work, Dr. Huber. We are most appreciative to have you here with us. Take it away.
Thanks, Rick. Let me share my screen. Sorry, getting there.
There it is.
Thanks for the introduction. In Australia, it's custom to actually acknowledge the country. So I would like to begin by acknowledging the traditional owners of lands on which we meet. These are the Ngunnawal people here in Canberra and all other traditional owners of traditional land, and pay our respect to past elders, present elders, and future elders. So we acknowledge that this is land, that it was, always will be traditional Aboriginal land. So thank you, Rich and John, for the very kind introduction, and also for Andy to invite me first. And as you probably already heard from the introduction, I'm a computational scientist by training, and I'm very grateful that you invite me anyway for this. So my group interest is really in algorithms, in tools, but we have also a long history in structural biology, in particular neurofocused in-cell structural biology. And then the other element we are doing is actually protein design, to use our computational tools to design completely new proteins. And about 6 years or thereabouts, we thought, well, maybe genetic code expansion is really what we need, because it not only allows us to install completely new functionalities as probe into proteins for structural biology, but also gives us new opportunities for protein design. And the other thing we thought, computational design should be very easily applied to genetic code expansion, because by the end of the day, all you have to do is actually make this orthogonal tRNA synthetase, which is, or should be, a very simple protein design. I mean, you know all how wrong we were, that this is not a simple computational design, but this how naive we were to get into this area. So maybe I can move my top bar so I see actually what is on the top. So today I want to talk to you about labeling of proteins or biomolecules, and everybody knows that labeling is the cornerstone of structural biology. And in particular, the challenges which are still left with labeling is, how can you precisely introduce labels into a biomolecule? How do you do this with very, very high yields? And then how do you make a method which is then transferable and easy to do so other people can take up the method which you can develop? So no surprise there, I will talk about genetic code expansion. I will briefly do nuclear spins, how we introduce nuclear spin labels into proteins. Then I will also talk to you about electron spin labels, how they can be introduced. I talk about reactive handles, and then do a side tour on how we actually also use reactive activated fluorophores as labels in protein. And then I very quickly wrap up the whole story. Okay, so for nuclear spin, fluorine is nearly the ideal nuclear spin which you can introduce to the protein. And there's many reasons for this. So one is fluorine is the only isotope. So fluorine-19 is the only isotope, is 100% abundance. It also has a very high gyromagnetic ratio, nearly as high as a proton. So therefore, your fluorine NMR will be extremely sensitive. But much more important is fluorine is a xenobiotic element, which means if you introduce this into a biomolecule, then you can do NMR on this biomolecule and get absolutely no background signals from normal elements. So this is really outstanding because you remove automatically all the background. Now, you have to deal with slightly broader line widths than if you do proton NMR. Fluorine NMR signals can be tens of hertz wide, but you also get a much larger chemical shift dispersion up to 200 ppm. And to show you this is just a couple of NMR spectra from different fluorinated amino acids. They are all aromatic fluorinated amino acids. And what you see is on the x-axis is the chemical shift, fluorine chemical shift, and the dispersion of the chemical shift is 25 or 30 ppm. So these signals are really widely dispersed over a wide range of ppm range, which means you get a relatively high resolution even if your line width is broad. Okay, so coming into genetic code expansion, just a couple of points of where our genetic code expansion might be a little bit different from what you're doing, other people are doing. So one of my favorite tRNA synthetase for genetic code expansion comes from this Archeon iso4G1. And we are generating a library which is relatively modest, about 27 million different variants, which are based on 7 sites which we mutagenicize, but they're not completely random mutagenicized, but they are a little bit rationally decided with the gene size. Now, the other thing which we introduced relatively early is we use a reporter, a red fluorescent amber stop codon interrupted red fluorescent protein as a reporter for amber stop codon suppression, and then use simply fluorescent activated cell sorting to sort out the functional active tRNA synthetases from non-active ones. So let me briefly show you just the principle on these fluoridated tryptophan. So this is 7-fluorotryptophan, what you see here in the middle, this molecule. And if you go through our selection system, these are outputs from fluorescent activated cell sorting. On the x-axis here, you see the relative red fluorescent intensity of each of the cells we analyze. So each dot is a single cell. On the y-axis, this is just the background fluorescence of the cell. You can ignore this axis for this purpose. So what you see is in our first round of selection, which is a positive round where we supplement the unnatural amino acid, we get about 0.4% of amber stop codon suppression, and we observe red fluorescence. I will come back on the 0.4% a little bit later, so keep this roughly in mind. So then we take this fraction and then we go through negative selection. Of course, when you leave the unnatural amino acid out, you want to have the negative selection selected. And then as you normally do through multiple positive and negative round of selections, we then finally of our initial library genetic pool, which are now selective, so which have selective but also specific tRNA synthetases for this unnatural amino acid. And then we can make monoclonal colonies to actually extract these tRNA synthetases. Now, does this work? So afterwards you take out the red fluorescent protein as your reporter. You do mass spectrometry. So here is a deconvoluted full mass spec of this protein. X-axis the mass, y-axis the intensity. And what you see is it doesn't have a single fluorotryptophan incorporated, but it has a second fluorotryptophan incorporated. Now, this is not because the gene has a second AMBA stop codon, but it comes from that the fluorescent protein originally has 3 other tryptophans, and one of these tryptophans can actually be replaced by fluorotryptophan. So the native tRNA synthetase for tryptophanyl tRNA synthetase can also accept fluorotryptophan as a substrate. Now, how do you overcome this? Well, if you add a lot of extra tryptophan, then you can actually suppress this extra incorporation. So then the tryptophanyl tRNA synthetase is saturated with normal tryptophan, accepts this substrate, and the engineered fluorotryptophan tRNA synthetase is actually highly specific that it still accepts the fluorotryptophan over the tryptophan. And you can prove this by just recording a fluorine NMR spectra. What you see is you get a single peak for a single substitution of the fluorotryptophan. It's not just randomly one of them is replaced by fluorotryptophan. So the reason why I point this out is, for me, this is still one of the miracles, or it's a very, very nice demonstration of how selective actually these enzymes can be, because if you look at the difference between a tryptophan and a fluorotryptophan, they are really tiny in terms of the molecule. If you look at the van der Waals radii of the fluorine versus the proton, fluorine is only slightly larger, similar, the bond length between the carbon and the proton or the fluorine is only slightly larger. So structurally, there's hardly any difference. There's only a slight polarity difference. So there's a polarity switch between the bond, but this small polarity switch is actually recognized by these mutants tRNA synthetases very, very selectively. Now, what can you do with these fluoridated tryptophans afterwards? Well, you can use them as very, very sensitive probes to probe conformational changes or changes which happen in your molecule. So we have shown this, for example, with this Zika protease. The Zika protease is the main protease from the Zika virus. It's essential for replication of the virus. It has 6 tryptophans, and what we did is replace each of these 6 tryptophans by fluorotryptophan in turn. So we have 6 different samples. And then you do an NMR spectroscopy on the 6 different samples. And what you see is there's 6 individual peaks, so 1 peak in each of the spectra. So it's very easy to assign what tryptophan you're dealing with. And then if you titrate an inhibitor of this Zika protease, which you see here on the top, you see shifts depending on how close or how affected the particular fluorotryptophan is by the proximity of the ligand. The other brief thing I want to point your attention to is on the right side here, I've noted down the yield from these fluorotryptophan proteins generated compared to the wild type. And roughly speaking, in this case, we get about 10% of the wild type yield, maybe 5% in this order of magnitude. And again, keep this in mind because a little bit later this will be important. But summary of this is you can really make proteins where you specifically change one atom, one proton to one fluorine in a whole protein of many atoms. Now, we've done a large variety of this fluoridated amino acid because they're such excellent probes. It's challenging for the selection, but quite rewarding. And we also have more recently done much more highly fluorinated amino acid is other mimetics for example, post-translational modifications. Okay, so these are spin labels. How about electron labels? And here on the right side, I show you a couple of sort of attachments of things which then introduce electron labels into your protein. So you have the classical nitroxide, which have an unpaired electron, so it gives you an electron label. Or the other ligands are actually chelating ligands, which can chelate, for example, lanthanides like a gadolinium, which have lots of unpaired electrons. So unfortunately, as you would imagine, we can't directly we haven't been able to directly genetically encode any of these large appendices in amino acids. But our solution to this is we use bioorthogonal reactive handles to attach these spin labels into a protein. And the particular bioorthogonal reaction we are keen for is this particular one, which we dubbed net-click reaction. We sort of serendipitously rediscovered it. We reported it, and then afterwards we discovered that this was already reported in a patent. But how it works is you have a electron-poor nitrile group, so for example, on a pyridine ring, a nitrile, and then this electron-poor nitrile can react with a amino-thiol group in a 1,4 orientation to form these thiazolines. And the reason why we really keen on this reaction is because it proceeds relatively rapidly. It proceeds in biocompatible conditions, but much more important, it doesn't require a catalyst. So you can really do this also in cells. So then we went on and actually genetically encoded cyanopyridine moieties. So here is cyanopyridine alanines, either in a meta position or a para position. And then you can demonstrate again that incorporated into proteins, they readily react and cleanly react with this not-click reaction. So down here is a mass spectrum of a small protein before the reaction and after the reaction, red. And you see it's a relatively clean reaction. Here it's relatively short. There's still some product, so some unreacted product, so substrate left. But the really important point about this reaction is you don't introduce any new chiral centers through the reaction, and there are also no regioisomers. And that's important for NMR or EPR applications because you want to have a single attachment of the attack and not have possible minor species of alternative attachments. Now, when we introduced this, the uptake was actually relatively slow because, like with many genetically encoded amino acids, the synthesis of these amino acids is hard, and people really don't want to invest a large amount of money to sort of custom synthesize natural amino acids to just try out this genetic encoding. So Elvi Abdelkader had a very good idea. He said, why can't we also biosynthesize the amino acids and then just start out with relatively cheap precursors, which everybody can buy? And he came across this system, which was reported in literature before. So this is cis-K-cis-M. This is two enzymes in all bacteria and many higher organisms. And it's a cysteine synthesis pathway to rescue cysteine synthesis. And it starts out with O-acetylserine and then can convert sulfide into cysteine. Now, other people have reported that if you supply thiol compounds, these enzymes can also convert these thiol compounds into amino acid. And then what Elvi has shown is that if you use these very simple cyano-thiol compounds, aromatic thiol compounds, we can easily make these amino acid here on the right side, which we call cis-CNP and cis-PIM for pyrimidine, because there's two nitrogens, directly in cell and only supply cheap precursors. So now that we can biosynthesize these unnatural amino acids, also we can combine this with genetic encoding. So then we have also genetically encoded these two unnatural amino acids to actually incorporate them into the proteins. So what this allows us now is making these proteins from the simple precursors and then use these as reactive handles to attach all sorts of things. Now, it's a nice biocompatible reaction, as I said, and just as a demonstration, if you do cell labeling, so this is labeling a outer membrane protein OMP-X with one of these cyanopyridine unnatural amino acid, you can label the outer membrane or the OMP-X molecule on the outer membrane, very simple with red fluorescent protein, if you put a cysteine at the N-terminus, because then you expose this amino-thiol moiety at the N-terminus of the protein. This can react to form these thiazolines and label the outer membrane. And this works actually fairly efficient, so you can go down to a relatively low concentration of your label, down to 20 micromolar of this red fluorescent protein, and you still get efficient labeling of your cells. So here is just cell sorting analysis again. On the top, the red part is after the labeling. On the bottom part, inverted in gray, is if you don't incorporate the unusual amino acid control cells in the cell, see there's a clear, strong labeling. And on the bottom is some microscopy picture, and something for me that really even E. coli cells, which are very small, the resolution is high enough that you clearly see the outline of the membrane where this labeling happened. Okay, the other sort of clever idea LV had is saying, well, if we have this reactive group which forms actually the thiazolines, maybe let's also try aromatic compounds, because aromatic compounds in this thiazoline reaction would make a much longer conjugated system. And he showed that this, what we call orgnet aromatic click reaction, is also relatively fast, because if you can do this, you can generate directly push-pull dyes under reaction on your protein. So using the different aromatic amino-thiol compounds, LV screened a lot. Here's a small panel of them. What you can do is reactively fluorescent label your protein, and you can fine-tune the properties of this push-pull tile in such a way that you get all sorts of fluorescent output, from blue fluorescence down to much more redshifted fluorescence. So these pictures down here are just photos of a normal mobile phone camera of a cuvette of the fluorescence, so there's nothing special. Now, just to make this point again, this is really reaction-activated fluorescence. So here on the right side, what you show is you have your protein and your precursor of a dye, which are both non-fluorescent, and really only on reaction, this moiety becomes much more conjugated and fluorescent. And on the left side, you see the fluorescent output. So the little dark black line on the bottom is the substrates, how fluorescent or what the fluorescence of the substrates is before the reaction. And you see there's a 50 times increase of the fluorescence under reaction in the fluorescent output. Now, the more redshifted you go, the higher the increase is, and you get more sensitive. Now, because of this high increase of the fluorescence, what you also can do now is actually wash-free fluorescent labeling of cells. We own this by incorporating this cyanopyridine-like amino acids in different proteins within a cell, and then do this reaction-activated fluorescent labeling. And then this is really only reactive labeling of the cells, but not washing the cells. So in the background, you still have the fluorescence of the unreacted substrates, and you see the unreacted substrates don't really add much to the background of your fluorescence. Okay, so hopefully I showed you that is a very easy way to incorporate these cyanopyridine moieties or pyrimidine moieties into the protein. How about the reactivity? Well, the reactivity is also sort of tunable depending on how aromatic or how electron poor you make your aromatic ring. So if you have a single nitrogen in the aromatic ring, the reactivity is roughly the same. However, if you add more nitrogens to the ring, the reactivity will go up. Now, how reactive is it? We've done kinetics. It's not super fast. It's probably on the top end of typically like strain-promoted click chemistry reaction, but it's by far not as fast as tetrazine click reactions, which many of you are familiar. And if you have any question to this, I have actually a backup slide which we can talk about. Okay, so, but here was our big surprise to it is when we did the biosynthesis of these amino acids, we had a big surprise of how well these amino acids can be incorporated. So if you looked at the yield, if the cyano, so the beta-cyanopyridine was incorporated, this was already a very highly efficient incorporation, and typically we get something like only 50% of the wild type yield of the expressed protein when we do the amber-stop codon expression. But when we do the same amber-stop codon suppression with this in vivo synthesized biosynthesized amino acids, what we observed is that the yield typically was equal or sometimes even higher than the wild type. And the only way we can explain this is really that the uptake of the amino acids is somehow limited, and these thiols compound have a much more active uptake and get converted very, very efficiently inside of the cells to the amino acid. But the outcome of this is really that you can incorporate these cis CNP or cis CNP PIM amino acids with very, very high efficiency and high yields within the cell. Okay, finally, let me also show you a couple of slides on spin labeling. So this is from collaborators at the University of Queensland, so Jeff Harmer and Vishal Bayer. They synthesized these gadolinium tags, which had an aminothiol group attached to it, so they can then be attached to proteins by this net click reaction. Now, there's lots of information on there. The only thing I want to point out is when you go to electron spin label is the electron magnetic moment is about 1,000 times higher than the nuclear moment. Therefore, you can go to much lower protein concentrations and lower sample volume. So in this case, we're talking about 50 micromolar protein in 60 microliter sample. And you get this wonderful Deer spectra. So Deer is where you measure the distance between two of these labels and measure then the distance distribution between two of these labels. Now, the big surprise for me of this was when they did the labeling of these ERP29 homodimer, they find very, very high labeling efficiency. So they claim 100% labeling efficiency for this labeling. And EPR is actually a good measure or a good way to measure labeling efficiency because you can quantitatively count how many spins are in your system, so how many gadolinium molecules are in your system, but also then by doing these Deer measurements where you do pairwise measurements, you can measure how many pairs are in this homodimer. So this means if your labeling efficiency of a single molecule was 90%, your Deer measurement would only see 81% of the Deer intensity. And therefore, you can really see exactly what the labeling efficiency was. Now, they have done this also with other sites on the protein, and they still find relatively high labeling efficiency if it's on the surface. So with this, let me briefly wrap up because my time is up. I hope I could briefly show you that we not only do computational work, but we also develop really useful tools, biochemical tools in genetic code expansion, which really allows precise, clean, and high yield labeling of proteins. And I hope I showed you a couple of magnetic resonance applications, which we hope to really push forward to characterize proteins into the native environment where we have low concentrations, but also do these measurements in cells. Finally, let me thank my relatively small group, but very active group, particular Hao Cheng, who does most of the genetic code expansion. He is really expert in this. Elvi Abdelkader is actually in Gottfried Ottwing's group, a very close collaborator, but does a lot of the protein expression work for us. And then, as I mentioned, Jeff Harmer at GQ doing the EPR spectroscopy. Now, last thing, thank you. And if there's any questions, I'm happy to answer them.
Thank you very much. That was excellent. I have questions, but I'll start with one that was posted in the chat. So this is from James Van Deventer. Says the discussion of the 7-fluorotrip incorporation selectivity was excellent. You're able to increase trip concentrations to millimolar levels to reduce fluorotrip incorporation at native trip sites. But have you had any success in reducing fluorotrip concentrations during your screens to identify synthetase variants that are more active towards fluorotrip or other fluorinated amino acids? And I'll add to that, did you stop at picking clones that were good enough? And do you think that if you would have continued your picking of clones, you would have found ones that were more selective?
I mean, absolutely. We always pick a small number of clones just out of practicality. But
Yeah.
I actually have a slide which might help in this. So we have not only done the 7-fluorotryptophan, but we also have done the 6-fluorotryptophan, 5-fluorotryptophan, and fluorotryptophan. And maybe this is a little bit too complicated, but this shows exactly the same data, but what you see here is we have picked a number of clones, different tRNA synthetases for each of the synthetases. They're all labeled with, let me start here on the right side, like F7 for 7-fluorotryptophan, and then they have some weird number, which are due to Hao Cheng's numbering. But you see, we picked a couple of clones, which we finally characterized. We picked much more than the 5 which are shown, but this is our initial clone picking. Have we picked enough? Probably not. We are not guaranteed that we find the optimal synthetase in our gene pool, but by picking 10, 20 of them, hopefully we find a good enough one. But what's more interesting about this graph is we also did the cross-specificity between the different picked clones for the selection of one synthetase round compared to the other substrate. So again, on the right side, the 7-fluorotryptophan synthetase come from a round of selecting for 7-fluorotryptophan, and then we tested them against the specificity for 7-fluorotryptophan, 6-fluorotryptophan, 5, and so on, and no unnatural amino acid. And what you see in the red colors is just the intensity of the red fluorescent, which develops when you do immunosuppression. Now, on this graph, what you see is, as you would expect for what you select for, has the highest red color. So if you select for 7-fluorotryptophan, the 7-fluorotryptophan is high, but there's also cross-specificity along to the other one. So for the 7-fluorotryptophan synthetases, they have cross-specificity with 6-fluorotryptophan to some extent. And if you pick the W36 mutant, you can incorporate both 6 and 7-fluorotryptophan. But they don't have cross-specificity to the other fluorotryptophan.
That's great. So in the interest of time, yep, go ahead.
John, can I ask one question?
Very quick one, because we have another one on the chat.
On this slide here, Thomas, amazing work on this slide. Do you know the concentration dependence of this sort of incorporation efficiency? Sort of, that's what is the concentration used in this particular scenario? And then if you drop it, how does it change? That's kind of what I was really excited. I know it's complicated and I know it's a ton of work. This is really, really exciting work and fantastic work.
So typically during the selection and typically protein expression, we just use the standard 1 millimolar of the natural amino acid. Now, we haven't really done a full concentration dependent yield and fluorescent measurements of how it changes with incorporation. But what we have done is, again, we are cheap researchers. Fluorotryptophans are not directly cheap to buy. Therefore, we typically go down to fluoroindols and then again biosynthesize the fluorotryptophan in cells. So you simply supply fluoroindol to the cells. This typically reduces the yield of incorporation because likely I assume that the fluorotryptophan concentration is lower in the cell. But we really haven't fully quantified it.
Okay. Really fantastic work. Thank you so much.
Really great. So there's one more quick question from Emerson Carmona. First off, very cool. Thank you. I'm interested in the physicochemical properties of your fluorophores. How bright are they? I'm curious to know about their quantum yields and extinction coefficients.
Lots of detail. Short answer to this is they're not super bright. But if I could refer you to our chem paper, which came out somewhere late last year or beginning this year, we've characterized them. We have quantum yields in there, and all the data you can look up. They are different depending on what reaction you do and what fluorophore you generate. But short answer is they're not super bright. They're not comparable to a Cy5, for example, because these conditions are still relatively small, and therefore the intensity of the fluorophore get high bright dyes. Now, having said this, we have actually done a Cy5 dye which conjugates to the same click reaction. It's a complete disaster because the Cy5 is so sticky and has the same fluorescent property as afterwards the reaction. So we react this with the cell, but then afterwards can't really wash out the substrate, and therefore it's a useless dye for labeling.